Good day, everyone. All right, class, starting here, we are entering the tribe Proteae, a very important group in Enterobacteriaceae diagnostics. So, when you hear Proteae, think of the ppm trio, comprising Proteus, Providentia, and Morganella. These organisms are widely distributed in the environment and also form part of our intestinal flora. Clinically, they behave as opportunistic pathogens. What immediately sets them apart from other Enterobacteriaceae? There are two key laboratory characteristics. First is that they are pod positive, meaning they can deaminate phenylalanine using phenylalanine deaminase. Second is that none of them ferment lactose. So you'll see non-lactose fermenting colonies on your McConkey agar. Whenever you suspect Proteae in cultures, pod positivity plus non-lactose fermenter profile is your biggest clue. Now, among the ppm group, Proteus is the most clinically recognized. So the two most important human pathogens are Proteus mirabilis and Proteus vulgaris. These are commonly isolated from urine, wound, ear, and even blood cultures. In urinary tract infections, Proteus accounts for about 3% in the United States. And what makes it more concerning is its ability to ascend the urinary tract, reaching even the kidney tubules. So this can lead to acute glomerulonephritis, especially in patients with anatomical issues or those with catheters. Also remember, Proteus is urease positive, and that urease activity contributes to stone formation. Let's talk about the stones, specifically strovite crystals. Because Proteus converts urea into ammonia, the urine becomes alkaline. When pH rises, magnesium and calcium phosphates precipitate and form strovite calculi, also known as infection stones. So these stones can grow large and fast, forming structures like staghorn calculi. So whenever a patient has recurrent UTI plus stones, always think of Proteus. One of the most iconic features of Proteus is its swarming motility on solid media. This happens because normal vegetative cells transform into elongated, hyperflagellated swarmers that move in coordinated waves. On culture plates, this produces concentric rings, which is a classic appearance. Clinically, this swarming ability is also linked to the organism's capacity to ascend up the urinary tract. Clinically, um, and in the lab speaking, most of the students or most of the scientists often describe the odor of your Proteus as burnt chocolate. So yes, even the smell is a clue. Next is a fun microbiology phenomenon, the genus line. If two different Proteus strains are swarming toward each other on an agar, they stop when they meet and form a line of inhibited growth. This occurs because of bacteriocins called proteasins or proteasines produced by different strains. So in research and epidemiology, this helps differentiate Proteus strains during outbreaks. Here, we compare Proteus mirabilis and Proteus vulgaris, in which Proteus mirabilis is indole negative, while Proteus vulgaris is indole positive. So both of them are pod positive, urease positive, and non-lactose fermenters. Remember this shortcut, everyone. Vulgar is positive, meaning Proteus vulgaris is usually indole positive. This distinction is routinely used in laboratories to identify species level. Just a quick historical note, Proteus antigens, specifically OX2, OX19, and OXK, cross-react with Rickettsia antibodies. So that's why Proteus was used in the whale Felix test, which is an old agglutination test for rickettsial infection. While not used much today due to poor sensitivity and specificity, you'll still encounter it in your immunology class. Moving on to Providentia, these organisms are non-lactose fermenters. They are pod positive, they are H2S negative, motile, and have a pungent odor. Unlike Proteus, Providentia species do not swarm. So they can ferment mannose, which helps in biochemical differentiation. There are five main species to remember. You have Providentia alcalifaciens, Providentia himbaceae, Providentia ritgeri. Providentia ritgeri is the only urease positive Providentia. You also have Providentia rustigiani. You also have Providentia stewartii. Among these, the clinically important ones are Providentia ritgeri and your Providentia stewartii. Providentia species can be isolated from urine, stool, blood, wound cultures, and even the throat. Clinically, Providentia ritgeri and Providentia stewartii are linked to human infections. Most common infection is nosocomial urinary tract infections. Providentia stewartii is specifically associated with UTIs in catheterized patients. Providentia can also cause skin infections, especially in burnt patients. Our last member of the ppm group is Morganella morganii. This is the sole species in this genus. The key biochemical traits of this organism are it is non-lactose fermenter, citrate negative, H2S negative, lysine decarboxylase negative, urease positive, and pod positive. This organism is opportunistic and can cause gastrointestinal infections, UTIs, and wound infections, particularly in immunocompromised or those on prolonged antibiotics. Although not as common as Proteus or Providentia, Morganella can cause serious infections such as septicemia, abscess formation, and post-operative infections. So it's also known for its multi-drug resistance, so treatment can be tricky in hospital settings. This table summarizes all the differential tests. When differentiating ppm organisms in the laboratory, remember Proteus, they are H2S positive in some species. They produce swarming. They are urease strong producers and are pod positive. Providentia, they do not have H2S production. There is no swarming and they are pod positive. For Morganella, they only have a simple biochemical profile such as citrate negative, urease positive, and pod positive. So these patterns are the most relevant traits to differentiate those three genus. Now we shift to the next genus, Citrobacter. These are normal gastrointestinal tract inhabitants and a common cause of hospital-acquired UTIs. There are three species commonly isolated from the stool: Citrobacter cosseri, Citrobacter braicaei, Citrobacter frondiae, and among the three, Citrobacter frondiae is commonly associated with disease such as UTIs, pneumonia, and intra-abdominal infections. It can even cause endocarditis in IV drug users. Citrobacter resembles Salmonella. So differentiation is really important. 80% produces H2S, which will result into black colonies. 50% are lactose negative. So in order to differentiate Citrobacter from Salmonella, urea hydrolysis and your lysine decarboxylation can help you differentiate the two. In urea hydrolysis, your Citrobacter frondiae, in which 70% of the strains are positive, while Salmonella is negative. In lysine decarboxylation, all of your Citrobacter species are all negative. So these two tests are your key differentiating markers. Alright class, let's move on to Edwardsiella, a small but interesting genus. These organisms are mostly isolated from cold-blooded animals, such as your reptiles, aquarium fish, frogs, and turtles. So if you see them in clinical specimens, you should already think environmental exposure or zoonotic link. Among the three species, between Edwardsiella tarda, Edwardsiella hoshinae, Edwardsiella ectaluri, only Edwardsiella tarda is significant in humans. So, Edwardsiella tarda is an opportunistic pathogen and can cause gastrointestinal infections, particularly gastroenteritis. So an important point, Edwardsiella can easily be confused with Salmonella and Citrobacter because they share some reactions. So, but here are the key biochemical traits. Urea hydrolysis, they are negative. Lysine decarboxylase, they are positive. Indole positive. H2S production, they are positive. Citrate negative. So this combination, specifically from what I have mentioned earlier, indole positive plus H2S positive is a classic biochemical characteristic of your Edwardsiella tarda. Next is Plesiomonas, which is used to be grouped under Vibrionaceae but was reclassified because it shares many features with Enterobacteriaceae. But take note, it's the only oxidase positive member of your Enterobacterales or Enterobacteriaceae family. Morphologically, they are motile with either a single polar flagellum or the so-called monotrichous flagella, or they can also have lupotrichous flagella, which appears in tufts of flagella. So Plesiomonas shigelloides is the only species in this genus. Plesiomonas shares several antigenic or biochemical traits with Shigella, but it's generally much less virulent. The most common clinical scenario is gastroenteritis after eating undercooked seafood, consuming untreated water, or exposure to contaminated fresh water. So think of this organism in patients with diarrhea after marine or freshwater exposure. When Plesiomonas infects humans, it can produce three types of diarrheal illness. The first one is watery or secretory diarrhea. This is the most common form. The second type is the subacute or the chronic diarrhea, which lasts 2 to 3 months. So this is more common in immunocompromised individuals. And the third type is the dysenteric form. Dysenteric form, it has blood and mucus. No? It can be present, which resembles the condition colitis. So yes, it can mimic Shigella, but again, virulence is milder. And here are the references used in this presentation. And with that, everyone, I would like to conclude the first part of our discussion under the family Enterobacterales or under the family Enterobacteriaceae. So thank you so much for listening and please watch out for the second part of this video. Thank you so much and once again, have a good day.